Our next speaker that we have lined up for us today is Dr. Yves Aubin. Dr. Aubin obtained his PhD in biophysical chemistry at Yale University and carried out his postdoc at the University of Toronto and Hospital for Sick Children. He later moved to the Merck Research Laboratories in Montreal, where he applied NMR spectroscopy to the studies of enzyme inhibitor interactions in support of medicinal chemistry. He then took a research scientist position at Health Canada, where he developed a research program in the characterization of protein structure of recombinant protein therapeutics. His laboratory is specialized in the production of isotope labeled proteins from E. coli to Pica pastoris in order to apply multidimensional NMR methods. This lays the basis for the development of analytical methods for the assessment of higher order structure of biotherapeutics and the study of excipient drug interactions. Without further ado, Yves, the floor is yours.
Thank you very much. Okay, now I can share. Coming. Here we go. Okay, now I should be in full. Thank you very much for the invitation. It's a real pleasure to share our latest results. And as you see from my introduction, since I joined the government, I have been applied to basically extend the use of NMR spectroscopy to the regulations of protein therapeutics, and in particular, these days, monoclonal antibodies, as you will see. So my goal in life as a chemist, I see proteins like basically large molecules. And to me, NMR can strongly contribute in bridging the gap between what we call now biosimilar and bring them closer to biogenerics. So coming from industry, and I guess all of you know that small drug therapeutics are very easily characterized mainly by NMR because NMR is an intrinsic part of their synthesis during their life cycles, and they can be exactly characterized. And that's why when they reach the end of their patents, generic drugs, copies, can be made and sold as basically a copies of the innovator. It's not the same though for biologics protein because the complexity of these molecules, the scaffold, and the way actually they are developed, they use an array of biological assays and various techniques. And it's interesting. I don't see all of my talks, but anyway, that's I'll try to guess what's in there because I know I wrote it. And the characterization of these molecules is very challenging. And because of that, we cannot say that this protein is actually the same because we only have indirect measurements of those proteins. And if you go back and how you basically discover a new protein, you are essentially blind. You use activity assays, cell-based assay, potency assay, and all sorts of assays that tells you that this band on a gel is your protein. You don't have in actual descriptions at atomic details of that molecule. So in the realm of regulatory science, we cannot talk about a biogeneric because we do not have a perfect characterizations of the molecular structure. And that's why the regulatory agencies have developed a biosimilar pathway to bring these follow-on product, as they used to be called, protein to the market, without having to do all the clinical assays and demonstration that this product is nearly similar to the innovator product. In particular, among these protein biotherapeutics, a class that have been drawing our interest for obvious reason are the monoclonal antibodies. At this time, there's about 1,000 monoclonal antibodies in the world at various stages of clinical development, among which there's over 200 that are under review or have been accepted as product. And the main reason for that is because these products, to summarize this slide, can be put into human very quickly. They are well understood. The technology to make them is very well understood and very, I would say, known by innovators. And the risks are low and the clinical trials are pretty straightforward. So they are basically the candidate drug that are the most rapid platform to bring to human and to treat any kind of disease. We have proposed a number of years ago using NMR natural abundance to have basically a 2D fingerprint of small cytokines like interferon and filgrastins. But what really opened the field is the basic, what I like to say, kicking the door open of MABs by my colleagues at NIST, where using their NIST MAB, they demonstrate that what we have proposed for smaller protein could be applied to large monoclonal antibodies. These molecules, as you will see, they're huge, 150 kilodalton, and thinking that we can get atomic descriptions using protein at natural abundance was also feasible for these proteins. So that was really a big barrier going down, but we have to go further and try to know more about these molecules, and I think NMR can bring us there. Whoops, the other way around. So here is a picture of what is a monoclonal antibodies. As I said, these are huge molecules, but what you need to remember for this morning is that a monoclonal antibodies is two polypeptide chains. The heavy chain, and I hope you see my mouse here, which is the heavy chain and the light chain, and two of those come together to form this structure. They are linked by a number of disulfide bridge. And on the right side, you see that these are adopting immunoglobulin fold. They are beta sheet, very stable fold. And one peculiarity of one of the subclass, the IgG1, is that they can be cleaved selectively with papain. So the Fab fragment, which is the antigen binding fragment, and the crystallizable fragment, referred to the FC fragment, can be separated. And these can be analyzed by NMR at very high resolution. But as you know, if we want to do an NMR, we want to be able to label and get atomic description, not just a pattern of signals. So we want to do labeling of these in order to apply the full power of NMR so that a complete resonance assignment can allow us to do a number of studies. We can monitor all sorts of perturbations, we can study dynamics, and we can get these atomic level descriptions that would be required if we want to call them one day biogeneric drugs, if we want to compare a subsequent product to an innovator. So we have been embarking on that endeavor since a number of years. We have explored the use of Pichia pastorbis, and our NIST colleague used our systems to basically apply to the NIST MAB, and they did a fantastic job. The challenge with Pichia, it's expensive in terms of the labeling, and the yields are not as much as you would like. We, in parallel, have explored the use of E. coli because E. coli, when it works, it can be pretty cheap. So to give you a bit of a summary here, when we want to label a protein, of that size, so a 50 kilodalton protein is the size of the Fab or the FC fragments, we want to use deuteration as well as glucose. And usually we will use glucose that is deuterated and C13 labeled to have all carbon-bound protons to be deuterons so that the 3D experiments that we use for backbone assignments will give us high sensitivity correlations so the assignment procedure can be done. And of course, we use N15 ammonium chloride, and cells are grown in deuterium oxide. If your protein is expressed as a soluble protein, you have a bit of a problem here because all the proton-bound nitrogen, the amide that we require to do the HNCACB and all these array of experiments that are N15 edited, we need to exchange back. Most of them will do so when they are in water, but a protein like a Fab or FC fragment is such a stable fold that they do not exchange even after a full month. And that's what my colleagues at NIST experienced from the solubly expressed NIST MAB producing yeast. They had to unfold the protein to allow this exchange. In addition, we would like to be able to assign and characterize the methyl on these side chains because methyls are very interesting probes that are basically very sharp, even for a large molecule like a full MAB. And having the assignment available for these can be very useful. There was a protocol developed by Lewis K. a number of 20 years ago, where if you use those two precursors, the atorbutyrate and ketoisovalerate, allows you to label the isoleucine delta 1 methyl as well as the valine and leucine methyl. So you don't have all of them, but you have a fair amount of those peaks that could be assigned, and then you get closer to a full characterizations of these large proteins. And these probes are very good reporter of protein dynamics as well. So not so long ago, we have proposed an approach where we use a single chain approach to make the Fab fragment. In other words, we express this polypeptide, which has the heavy chain part of the Fab connected with a linker that is removable with the light chain. We express that in E. coli. We make large amount of it because E. coli can make garbage very efficiently because that goes in the trash can, namely inclusion bodies. The beauty with that, we don't have the issue of back exchange because as soon as we start refolding this polypeptide chains, we have our amide proton back. We have this sort of a complex molecule with the linker in a histag. The histag was originally put there so we can do on-column refolding. That worked, but unfortunately, the efficiency of refolding was not that good, so we went into a bucket type like a fast dilution protocol that we have described. And upon cleavage, we get these Fab fragments. This approach worked for 4 out of the 6 that we have originally tried, bevacizumab and infliximab. One expressed very highly, but the refolding was not very efficient. So we knew we had some homework to do to maybe optimize the linker. And also cleavage of the linker was very, very low yield. Somehow papain used to cleave the linker was destroying 80% of our proteins. So that became a costly enterprise. So we sort of looked into a different approach, something we have tried to stay away. In other words, having the 2 chains brought together during the refolding. But before going there, I'm getting ahead of myself just to show that this approach did produce well-folded Fab fragment. On the left panel, the A panel, this is adalimumab that is overlay with the natural abundance spectrum of Humira. As you can see, it's a pretty good match. So we have refolded this E. coli materials the same as the drug purchased at the pharmacy. And same for trastuzumab, but this time using the metal spectrum to show also a different reporter of the well-folded protein made in E. coli. We managed to proceed in doing the resonance assignments. This is trastuzumab that was done by Donald Gagné, but in parallel, adalimumab had been assigned by Mouzadid. This endeavor takes about 3 months, 4 months. It's a very, very complex puzzle to solve, but can be solved with all the NMR techniques that we can apply. These are complex puzzles. We have the light chain and heavy chain, and you have about 440 resonances. By using 3D NMR, we can achieve that. But I was not satisfied with that. And again, I'm getting ahead of myself. Using the precursor as I described, we can assign those methyl groups very efficiently using those 2 experiments. We correlates the methyl groups to the NH either via the carbonyl or via an HCA transfer that allows both the amide of the I residue and the previous ones to connect, which is sometimes helpful when you have some ambiguity in your assignments and get most of these assigned. So we were not happy with a number of things. It's a very complex puzzle. It takes a lot of time, many months to get the assignments. Our colleagues at NIST had the same challenge with the NISMAB, and you get about over 90% of the backbone assigned. But we wanted to know, can we do this better? One big problem that we have with our approach is that the single chain approach, the linker, was a problem. So what we did is we decided to follow on some of the previous work where they used the 2 heavy chains of the Fc fragment and they on-column refolded, and they were able to produce protein and refold it and assigned it. So we did a modified protocol, and we're able for the Fab fragment, and we're able to get very efficiently 300 to 400 micromolar NMR samples with only half a liter. With this new approach, we're able to basically get the Fc fragments. And the beauty with this, we only have the equivalent of 200 residue. The assignment in this case, de novo, we didn't look at the previously reported, we're able to do that within a couple of hours at about 80%. So we went on, so basically learning from this, we went with the Fab fragments. So I need to accelerate so I don't run out of time. And we're able to do bevacizumab, infliximab that didn't work with the previous protocol, and rituximab as well. But since we're mixing the two chains, we thought, why not simply label one chain prior to refolding and using the other chain not labeled? And we're able to get subspectra of the heavy chain in blue and the light chain in red and assign them. So that approach allows us to assign these things not in 3 months, but in less than 3 hours, basically in about an hour. Basically a walk in the park because there is no overlap that are difficult to unravel. I'll give you an example here where you have bevacizumab and you have this little square here that is represented there, and you have in blue and red the light and heavy chain assignments. And to point here, you have 2 amide on top of each other. So you have 6 carbon correlation coming in 12 in total, in fact, because you have these 2 overlapping that are impossible to resolve. So that is a bottleneck in the assignment. In this thing, there's basically no overlap. So assigning this bottom panel left and right takes about an hour. This takes months. So that was a great, great, great improvement. And one thing we found out, and I'm going to conclude with that quickly, somehow, when Donat was preparing sample, he didn't use the doubly labeled glucose because he was making many, many tests. He went out with C13 glucose only. And what happened is that we found out we can still do very high backbone assignments very quickly. In fact, everything I showed you is coming from only using C13 glucose. Deuterium oxide is used. But one thing we find out when we're analyzing the metal groups spectrum is that not only we can get the ILV and isoleucine delta 1, but we could also get the alanine, the threonine gamma 2, and the isoleucine gamma 2, which was a big bonus. And here, what I mean here. This is the HSQC of a sample of 20%, and you can see that most of them can be assigned. On the left, on the middle and right panel, you have the ILV, triply labeled bevacizumab, either using C13 glucose only or doubly labeled. Let's look at the doubly labeled glucose, and this is basically a typical ILV metal signature. And look at the extra resonances that you have. So if we look, we see the same thing for the light chain on the right. And if we keep going, we see we have this funny shape. It's because the metals are basically going through incomplete deuteration in the biosynthesis pathway. When a proton becomes exchangeable, it can be exchanged with the deuterium from the solvent or the media. And we have these isopotmers. So we have the CH3 and CHD2 and CH2D and CHD2. But the right assignments will be the one with the CH3. And you can see on the right panel that the alanine, the CH3 is very, very, very weak, but we can still assign them unambiguously. And of course, we can get that also for the FC fragment as well. So in summary, using this approach, what we call differential labeling, in 1 hour, one pass, not going and digging through the data, the light chain, we can nearly get everything assigned. On top of that, we can assign almost all of the metal groups. This is also true with the Fab, but the Fab has an interesting behavior I don't have time to discuss. We have slightly lower, but still. And this is not going through using any 40 experiments, the basic 6 experiments that you can get off the manufacturer. You press go, you get the data, and you analyze them. In comparison, and I'll finish with that, the doubly labeled glucose gives you slightly superior statistics because you have full relaxation, and some correlations that were weak and could not be assigned readily on the first pass could be done more efficiently on that first pass. And of course, you're missing some of the metal groups. That procedure, from the time you put your DNA into your cell, when you do a fresh transformation, your culture, to the end you get those data, takes less than 6 weeks. So with no further ado, I will basically thank all the people that contributed to that, especially Donald Gagné, whose work I presented this morning, and Mouzadid, and of course, other members of my lab and my colleagues who helped in characterization those proteins the first time we did a single chain. Of course, my longtime collaborator, colleagues at NIST and at the NRC, where we used the 900 megahertz for some of the data, and of course, Louis K for providing pulse sequences for the sidechain experiment. And finally, thank you for your attention.
Wonderful. Really excellent work. Very impressive, especially on the timing of everything too, and how quick you're able to do the sampling. We do have a question in the Q&A box. Would it be an option to work with metabolic precursors, selective labeling with alpha ketobutyrate and alpha keto isovalerate that harbor an isolated spin system, the 13CH3 next to carbon 12 that are deuterated, in order to avoid expressing in D2O and subsequent refolding?
So we do refolding in these things. The protocol is expressing inclusion bodies, and we refold the protein. We're not expressing soluble protein here. That's one thing. The second thing is any fancy you have in selective labeling and whatnot is possible. Because you are using E. coli, you are expressing inclusion bodies. So if you have an oxotrophic strain of E. coli, we're using the BL21DE3, but you have another one, and you can produce any kind of labeling schemes that you desire. Once you have the inclusion bodies and you apply our protocol, which we will publish shortly, the paper is written, waiting internal review to send it out, you can do that. So it's economical, it's quick, it's fit, and it's cheap. Because all the samples we've shown of these various fab, they were at 300 to 400 micromolar protein concentration. And no more than 1 liter of culture of each chain was required. In most cases, only half a liter, and we had those. In fact, when we did 1 liter, we had to split the sample in 2 because we had too much protein. We didn't want to go much higher than 500 micromolar. And the quality of the spectrum, the intensity of the correlation is excellent. So the assignment was very, very easy.
Awesome. Thank you very much. We're going to have to move on to the next speaker, but there is one more question in the Q&A box. If you're able to go ahead and answer that, that would be wonderful.
Thank you.
Thank you very much. Excellent talk once again.